Okay, all of you, good morning, students. Okay, my name is Ashok. I am senior lecturer in botany. Okay, now today discuss about that the respiration in higher plants. Okay, in respiration in higher plants, aerobic respiration, aerobic respiration, that is step 2. Step 2. Okay, right. In previously we discussed the step 1, that is glycolysis. The step 2 is Krebs cycle. Okay, next step 3 is electron transport system. Okay, today I am discussing about that aerobic respiration. Step 2 in plants. Okay, that is Krebs cycle. That is Krebs cycle. Okay, Krebs cycle. This Krebs cycle is also known as citric acid cycle. Citric acid. Citric acid cycle. It is also known as tricarboxylic acid cycle. Tricarboxylic, that is TCA. Okay, tricarboxylic acid cycle. Okay, one by one, we briefly explanation of these definitions. Okay, first of all, Krebs cycle. In plants, the respiration process, the first time proposed by Sir Krebs. Sir Krebs. So, it is referred as a Krebs cycle. Referred as a Krebs cycle. Right, next terminology, that is citric acid cycle. Okay, in aerobic respiration step 2, Krebs cycle, the primary stable product in this pathway, that is citric acid. That is citric acid. The citric acid is a 6-carbon compound. Okay. In this pathway, the primary stable product is citric acid, so it's called citric acid cycle. Okay. It is also known as a tricarboxylic acid cycle. Okay, the tricarboxylic acid means the primary stable product in a Krebs cycle, that is citric acid. This citric acid is composed 3 carboxylic groups. 3 carboxylic groups. Okay, so 3 is means tricarboxylic groups. So carboxylic acid cycle. Okay, Krebs cycle. Sirhana Krebs proposed first time, so it's called Krebs cycle. Citric acid cycle. In this pathway, the primary stable product is citric acid. It is a 6-carbon compound, so it's called citric acid cycle. Then tricarboxylic acid cycle. Tricarboxylic acid cycle means the primary stable product citric acid is 3 carboxylic groups, so it's called tricarboxylic acid cycle. Okay, right. Next, here how to learn this Krebs cycle. అసలు ఎందుకు నేర్చుకోవాలి ఈ Krebs cycle వల్ల ఏంటి యూజ్ అనేది ఒకసారి చూస్తున్నానా. రైట్, in the plants the respiration takes place. Okay, that is aerobic respiration. Okay, in this pathway, the complete oxidation of food, complete oxidation of food material, the complete oxidation of food material, then it yields yielding CO2 and water. CO2 and water. CO2 and water. Okay, right? In plants and in organism, okay, the complete oxidation of food takes place in aerobic respiration step 2. Okay, then resulting the formation of CO2 and water is released. Okay, so this citric acid is main importance. The liberation of energy, liberation of energy, the production of ATP, production of ATP. Right. Next, this Krebs cycle where it takes place. Okay, the site of Krebs cycle, that is mitochondrial matrix. Okay, in previous class we discussed the step number 1, glycolysis. The glycolysis takes place in the cytosol of the cell. Okay, right. But the step number 2 in aerobic respiration Krebs cycle takes place in the mitochondrial matrix. Okay, already we know the mitochondria, it is powerhouses of cell. Okay, right. Next, in this pathway, in this Krebs cycle, the total chemical reactions takes place 10. The total number of chemical reaction, that is 10 chemical reactions. 10 chemical reaction. Okay, the food is completely oxidation, then yielding CO2 and water with the help of the 10 chemical reaction takes place. Okay, this out of 10 chemical reaction, the 4 chemical reactions are, okay, the 4 chemical reactions are oxidation. Okay, the 4 oxidation steps takes place in out of 10 chemical reactions. 10 chemical reaction. Okay, right. Then overall this Krebs cycle is involve 10 chemical reaction. Okay, right. So this 10 chemical reaction we discuss briefly in this topic. Okay, one by one. Right. The first chemical reaction, that is condensation. Okay. The first chemical reaction in Krebs cycle, condensation. Okay. Second chemical reaction, dehydration. Okay. The third one, hydration. Next, fourth one, oxidation. Oxidation 1. Okay. Total chemical reaction, 10 chemical reaction. Out of 10 chemical reaction, the 4 reactions are oxidation. So the first oxidation, 4th step. Next, 5th one, decarboxylation. Next, 6th chemical reaction, oxidation 2. Here in the 6th chemical reaction, total 3 reactions takes place. The first one is oxidation. Okay. The second one is decarboxylation. Then the third one is condensation. Okay. In only 6th chemical reaction, the oxidation, decarboxylation and condensation takes place. So overall oxidation, decarboxylation and condensation. Okay, the seventh chemical reaction in Krebs cycle, that is cleavage. Cleavage. Okay, cleavage. Right. Cleavage, also known as SLP, substrate level phosphorylation. Okay, the cleavage SLP3, it is very important. Okay, already in previous class we discussed in glycolysis. Okay, the SLP1 and SLP2, that is seventh chemical reaction, dephosphorylation, and the 10th chemical reaction again dephosphorylation. The first seventh chemical reaction dephosphorylation is SLP1. Next in glycolysis 10th chemical reaction dephosphorylation 2. Dephosphorylation, that is SLP2. Right? What is SLP? SLP means substrate level phosphorylation. Okay, that is expanded name, substrate level phosphorylation. Right? It means the direct synthesis of ATP. The direct synthesizing of liberation of energy in the form of ATP. So, it's called substrate level phosphorylation. Substrate level phosphorylation. Okay, right? In this cleavage, also two reactions are takes place. Okay, number one, cleavage SLP3. Okay, next hydration. Okay, then next decondensation. Right. Next, eighth chemical reaction. Again, oxidation 3. Okay, it is single only. Right. Ninth chemical reaction, that is hydration. Ninth chemical reaction, that is hydration. Okay. Then the last chemical reaction in this pathway, that is oxidation number 4. Oxidation 4. Right. This is 10 chemical reaction which help for the complete oxidation of food and yielding CO2 and water, and also the liberation of energy in the form of ATP. Okay, one by one, this chemical reaction definition also I explain. Okay, the first one is condensation. The condensation means addition. Okay, here in this pathway, the Krebs cycle start with the acetyl coenzyme A. This acetyl coenzyme A is a 2-carbon compound. Okay, this 2-carbon compound acetyl coenzyme A is condensed with the addition with the oxaloacetic acid. That oxaloacetic acid is a 4-carbon compound. Okay, then the resulting the formation of the primary stable product, that is citric acid. Okay, so it's called citric acid cycle. Okay, the citric acid is composed 3-carboxylic group, so it's called tricarboxylic acid cycle. This total pathway is proposed Sirhana Krebs, so honorably it also it's called Krebs cycle. Okay, right. Condensation means addition. Okay, the smaller compounds are addition, then form larger compound. So that is condensation. Okay, it's clear now? Right. Next, dehydration. Okay, hydrates, it means water. Okay, D means removal or eliminated. Okay, the eliminated of water or removal of water, so that is known as dehydration. Next, third chemical reaction, hydration. Hydration means water addition of water. Addition of water, so that is hydration. Next, oxidation 1. Okay, the oxidation means the gain of oxygen. Okay, here the gain of oxygen. Okay, and loss of electrons and protons. Electrons and proton, so that is oxidation. The gain of oxygen and loss of electrons and protons, okay, that is oxidation. Overall, in this Krebs cycle, the 4 oxidation steps are takes place. That is 4th chemical reaction, next 6th chemical reaction, 8th chemical reaction, and 10th chemical reaction. Okay, this 4 oxidation takes place in this pathway. The oxidations means the gain of oxygen and loss of electrons and protons. Okay, then 5th step, that is decarboxylation. Okay, D means removal or eliminated. Which are removal here? Carbon dioxide is removal. The removal of carbon dioxide is known as decarboxylation. Right? Next, here condensation repeated, decarboxylation repeated, oxidation also repeated. So that is 6th. 7th one, cleavage. Cleavage. The cleavage is nothing but substrate level phosphorylation. Substrate level phosphorylation means, already we discussed, the direct synthesizing of ATP in this pathway. Okay, the direct liberation of energy in the form of ATP. So that is substrate level phosphorylation. Hydration already here. Hydration means addition of water. Decondensation means, D means removal or eliminated. Okay, condensation, the compound are eliminated. Here the smaller compound are addition, then form larger compound, that is condensation. Okay, in this large compound, the smaller compounds are eliminated. Here, particularly the coenzyme A. The coenzyme A is eliminated in this step. Okay, so that is decondensation. Right. Next, 8th one, oxidation. Oxidation already we discussed. Next, hydration. Oxidation 4. Okay, here the mainly entrance in a NEET and MSET, the site of Krebs cycle, that is mitochondrial matrix. The Krebs cycle is also known as tricarboxylic acid cycle. Why? The primary stable product citric acid is composed 3 carboxylic group is known as tricarboxylic acid cycle. Okay, then how many chemical reaction takes place in this pathway? 10 chemical reaction. Okay, what are the steps of oxidation in Krebs cycle? Okay, that is 4th chemical reaction, 6th, 8th and 10th. This 4, 6, 8, 10th are oxidation steps in this pathway. Okay, right. The Krebs cycle begins with the 2 carbon compound. The 2 carbon compound, that is acetyl coenzyme A. Acetyl coenzyme A. Right. The Krebs cycle begins or start with the acetyl coenzyme A with condense the 4 carbon compound, that is oxaloacetic acid. Oxaloacetic acid. Okay, that is 4 carbon compound. Okay, this 4 carbon compound and 2 carbon compound is linked, then form 6 carbon compound, that is citric acid. That is citric acid. So it's called citric acid cycle. Okay, right. This is Krebs cycle, the smaller introduction about Krebs cycle. Then next, the total chemical reaction one by one we discuss here. Okay, right. First here, in Krebs cycle, the begins with the two carbon compound, that is acetyl coenzyme A. This acetyl coenzyme A are already formed in the step 2, link reaction, that is okay, that is link reaction step 2. Okay, step 2 is a link reaction. Okay, this is step 3 now. Krebs cycle is a step 3. Okay, the small mistake. In aerobic respiration, totally 3 steps. Okay, the first one is glycolysis, second one is link reaction, the third one is Krebs cycle. So it is rectified. Okay, next one by one we discuss in this topic. Okay, the acetyl coenzyme A already we discussed in previous topic. Okay, link reaction step 2. The step 2 link reaction, the finally resulting the formation of acetyl coenzyme A. Right? This acetyl coenzyme A, this acetyl coenzyme A, it is 2 carbon compound. It is resulting in the step number 2 link reaction. Okay, right. This acetyl coenzyme A is condensed with the oxaloacetic acid. Oxaloacetic acid. This oxaloacetic acid is a 4 carbon compound. 4 carbon compound. It is very important carbon number, 4 carbon compound. Okay, oxaloacetic acid. Okay, right. This 2 carbon compound acetyl coenzyme A, they condense with the 4 carbon compound oxaloacetic acid. Then finally resulting the formation of citric acid. Citric acid. This citric acid is a 6 carbon compound. 6 carbon compound. In this step, one of the enzyme is required. This enzyme is known as synthase enzyme. Okay, synthase enzyme. Synthase. This is synthase enzyme required here. Okay, this is first step in the Krebs cycle. This is first step. Okay, what is the first step? Condensation. Okay, the 2 carbon compound of acetyl coenzyme A is condensed with the 4 carbon compound of oxaloacetic acid, then resulting the formation of 6 carbon compound, that is citric acid. In this reaction, one of the enzyme is required, that is synthase, citrate synthase enzyme. Okay, right. Then second step in a Krebs cycle, that is dehydration. Okay, this okay, the mole number also very important in this topic. Okay, in step number 2 aerobic respiration, step number 2, the 2 moles of acetyl coenzyme A, the 2 moles of acetyl coenzyme A is released. This 2 moles of coenzyme A, acetyl coenzyme A is condensed with the 2 moles of oxaloacetic acid, then resulting the formation of the 2 moles of citric acid. Okay, the 2 moles of citric acid is synthesized here. citric acid. This citric acid is a 6 carbon compound. Okay. Next, dehydration. This 2 moles of citric acid, then removal of water. Okay, here the 2 moles of water is removal. The 2 moles of water is removal. So that is dehydration. Then resulting the formation of cis-aconitic acid. Cis-aconitic acid. Cis-aconitic acid. Okay, this cis-aconic acid also 6 carbon compound. 6 carbon compound. Okay, here one of the enzyme is required, that is aconitase. Aconitase, right? The aconitase enzyme required here. Aconitase enzyme required. This is second chemical reaction in the Krebs cycle. Okay, right. Next, third chemical reaction, that is hydration. The hydration means addition of water. Okay, the 2 moles of water is addition to the 2 moles of water is addition to the cis-aconic acid. Okay, that resulting the formation of 2 moles of isocitric acid. 2 moles of isocitric acid. This 2 moles of isocitric acid, okay, their carbon number also very important here, that is 6 carbon compound. Right. Here also one of the enzyme is required, that is again aconitase. Again aconitase enzyme required here. Okay, right. This is third chemical reaction in the Krebs cycle. Third chemical reaction in the Krebs cycle. Okay, right. Next, fourth chemical reaction. Fourth chemical reaction, that is oxidation. Okay, right. This 2 moles of isocitric acid undergo oxidation. Undergo oxidation. It means the 2 moles of NAD plus. Okay, here the NAD means nicotinamide adenide. Okay, nicotinamide adenide diphosphate, that is NADP, but here nicotinamide adenide di. Okay, so this is 2 moles of NAD. Then convert into the 2 moles of NADH2. NADH2, that is NADH plus H plus. Okay, then resulting the formation of oxalo. Okay, oxalosuccinic acid. Oxalosuccinic acid. How many moles oxalosuccinic acid is formed? Okay, here the enter of, okay, beget of Krebs cycle, the 2 moles of acetyl coenzyme A. Okay, so entire in this cycle the 2 moles of products are formed. Okay, right. 2 moles of oxalosuccinic acid. 2 moles of oxalosuccinic acid. Here also very important. Already we discussed the mole number. Okay, the mole number of products and carbon number of products. It is very important for objective. So the oxalosuccinic acid also 6 carbon compound. 6 carbon compound. Here, one of the enzyme is required. One of the enzyme is required. That is dehydrogenase. Dehydrogenase. Dehydrogenase enzyme. Dehydrogenase enzyme. Okay, right. Here, this is a fourth chemical reaction. Fourth chemical reaction. Right. Next, fifth chemical reaction, that is decarboxylation. Right. These 2 moles of oxalosuccinic acid is removal. The 2 moles of oxalosuccinic acid is removal. 2 moles of CO2. 2 moles of CO2. Okay. The 2 moles of CO2 is removal. Okay. Then that is known as decarboxylation. Then finally resulting the formation of alpha ketoglutarate acid. Okay. Alpha ketoglutarate acid. Alpha ketoglutarate acid. Okay, full form alpha ketoglutarate acid. Alpha ketoglutarate acid. Okay, right. Here one of the enzyme required, that is decarboxylase. What is that? Decarboxylase. Decarboxylase enzyme utilized in this fifth step. This is fifth step. Number 5. Fifth chemical reaction. Okay, right. Next, sixth chemical reaction we discuss. Okay, right. This 2 moles of alpha ketoglutarate acid. Here the carbon number is must and should. Okay, then the alpha ketoglutarate acid is 5 carbon compound. 5 carbon compound. Okay, right. These 2 moles of alpha ketoglutarate acid undergo oxidation, condensation, and as well as decarboxylation. Okay, these 2 moles of alpha ketoglutarate acid undergo oxidation. The oxidation already we discussed. The 2 moles of NAD plus is converted into 2 moles of NADH plus H plus, NADH2. Okay, this is oxidation. Okay, in the sixth chemical reaction, the total 3 chemical reaction takes place in single sixth chemical reaction. First one, oxidation. Okay, oxidation number 2. Okay, condensation. The condensation means the 2 moles of coenzyme A. The 2 moles of coenzyme A is condensed to the alpha ketoglutarate acid. So that is a condensation. Okay. Next, then third one, decarboxylation. Okay. Decarboxylation means already we discussed in fifth chemical reaction. Okay, the 2 moles of CO2, the 2 moles of CO2, right? This is arrow marks, very very important. This 2 moles of CO2 is removal from alpha ketoglutarate acid. Then finally resulting the formation of succinyl coenzyme. What is that? Succinyl coenzyme A. Succinyl coenzyme A. Okay, this succinyl coenzyme A, the carbon number is very very important. That is 4 carbon number. 4 carbon number. Succinyl coenzyme A. How many succinyl coenzyme A form here? The 2 moles of succinyl coenzyme A form. Okay, this coenzyme A is added to succinic acid. So that is succinyl coenzyme A. Right? Here one of the enzyme is required, that is same case dehydrogenase. What is that? Dehydrogenase. The dehydrogenase enzyme utilized in this sixth chemical reaction. Dehydrogenase. Dehydrogenase. Okay, right. Next, seventh chemical reaction. The seventh chemical reaction, what is that? Cleavage. In the seventh chemical reaction also 3 chemical reaction take place. Number 1, cleavage. Okay. Number 2, decondensation. Here the coenzyme A is addition, so that is condensation. Here the coenzyme A is removal, so that is decondensation, and as well as hydration. And as well as hydration, it means addition of water. Right? These 2 moles of succinyl coenzyme A is undergo cleavage. The cleavage means here the 2 moles of ADP, adenosine diphosphate, then the 2 moles of inorganic phosphate is combined, then form 2 moles of ATP. 2 moles of ATP. Right? This cleavage is also known as substrate level phosphorylation 3. Okay, means the direct liberation of energy in the form of ATP. So that is substrate level phosphorylation. Okay, it is one complete, cleavage complete. Then second, in this reaction, that is hydration. Hydration. The 2 moles of water. The 2 moles of water is addition to the 2 moles of succinyl coenzyme A, and as well as the 2 moles of coenzyme A. The 2 moles of coenzyme A is removal, eliminated. So eliminated from succinyl coenzyme A, then resulting the formation of succinic acid. What is that? Succinyl. From succinyl coenzyme A, the coenzyme A is a removal, decondensation. So remaining which product is forming here? Succinic acid. That is succinic acid. Succinic acid. The succinic acid, the carbon number is 4 carbon. As it is 4 carbon. How many moles are liberated succinic acid? Same case, the 2 moles. Each product is liberated 2 moles. Okay, 2 moles of succinic acid. Right? Here this is 6th chemical reaction. 6th chemical reaction. Here this is 7th chemical reaction. Okay. In 7th chemical reaction, one of the enzyme is required. Okay, that is succinic thiokinase. It is very very important enzyme. Okay, it is pacemaker enzyme. Okay, right? Succinic thiokinase. Succinic thiokinase. Okay, simply we write succinic లేకున్నా పర్లేదు, థయోకైనేజ్ అని పెట్టినా నో ప్రాబ్లం. ఓకే, థయోకైనేజ్. ఓకే, రైట్. నెక్స్ట్, 8th chemical reaction. This 8th chemical reaction is oxidation. Oxidation. These 2 moles of succinic acid undergo oxidation. Here the 2 moles of FAD plus converts into 2 moles of FADH2, 2 moles of FADH2. So that is oxidation 3. Okay, what is the difference between here in fourth chemical reaction NAD place, in sixth chemical reaction NAD place? Okay, these are also oxidations, but here the instead of NAD place, then transfer, okay, then transfer FAD place, flavin adenine dinucleotide, so that is FAD place. Okay, right, the NAD place and FAD place, NADH2, FADH2, the 1 mole of NADH2 is equal to 3 ATPs, the 1 mole of FADH2 is equals to 2 ATPs. Okay, this we briefly learn the next topic, that is balance sheet. Okay, in total number of ATP production in aerobic respiration. Okay, balance sheet next class we discuss. Okay, right. Okay, simply remember the NADH2 is equal to 3 number of ATP, FADH2 equals to 2 number of ATP. Okay, NADH2, FADH2 2 ATPs, FADH2, NADH2 3 ATP ki equal. Okay, ఇది మనం మళ్ళీ బ్యాలెన్స్ షీట్లో వస్తుంది ముందు చాప్టర్. ఓకే, ఏరోబిక్ రెస్పిరేషన్లో ద టోటల్ నంబర్ ఆఫ్ ఏటీపీ ప్రొడక్షన్ బ్యాలెన్సింగ్ మనం చూపిస్తాం. ఓకే, అది నెక్స్ట్ టాపిక్లో మనకి FADH2, NADH2 ఈక్వల్ టు ఏటీపీ, NADH2 3 ATPకి ఈక్వల్, FADH2 2 ATPకి ఈక్వల్. ఓకే, రైట్. These 2 moles of succinic acid undergo oxidation, that is oxidation number 3. Okay, must remember in oxidation 1, in oxidation 2, in oxidation 4, the NAD plus convert into NADH2, but in oxidation 3, instead of NAD plus, okay, then join FAD plus. This FAD plus is converted into FADH2. Okay, right. Here one of the enzyme required, that is dehydrogenase. Okay, right. Then resulting the formation of fumaric acid, the 2 moles of fumaric acid is formed. 2 moles of fumaric acid is formed. 2 moles of fumaric acid formed. This fumaric acid also 4 carbon compound. Okay, in the previous need year question paper, the carbon number is asking carbon number. Okay, so must remember the moles of products and carbon number of products. Okay, right. This is 8th chemical reaction. The 8th chemical reaction is complete. Okay, next 9th chemical reaction in Krebs cycle. 9th chemical reaction in Krebs cycle. Okay, right. That is hydration. What is that? Hydration. The hydration means the 2 moles of water is addition. 2 moles of water is addition. The 2 moles of water is addition. Okay. The 2 moles of water is addition to the fumaric acid, then form malic acid. What is that? Malic acid. 2 moles of malic acid is formed. Malic acid is formed. Okay, here one of the enzyme required. Okay, malic acid carbon number also 4 carbon number. 4 carbon number. Okay, right. Next, here one of the enzyme require here, that is fumarase enzyme. What is that? Fumarase enzyme. Okay, this is ninth chemical reaction. Ninth chemical reaction. Okay, right. Next, last one, 10th chemical reaction in Krebs cycle. That is oxidation 4. Okay, the 2 moles of NAD plus is converts into the 2 moles of NADH2. 2 moles of NADH2. Okay, this 2 moles of malic acid is undergo oxidation, then finally resulting the formation of 2 moles of oxaloacetic acid. Okay, here the cycle is continuously takes place. Okay, then yielding of CO2 and water, and as well as liberation of energy in the form of ATP. Okay, right. మరి ఇది ఐపీకి ఎలా గుర్తుపెట్టుకోవాలి? ఓకే, ఆబ్జెక్టివ్కి ఇన్ అండ్ అవుట్ ఏ ప్రోడక్ట్స్ ఇక్కడ రిక్వైర్ అవుతాయి, ఏ ప్రోడక్ట్స్ రిలీజ్ అవుతాయి అనేది ఒకటి గుర్తుపెట్టుకోవాలి. అది కూడా నేను మళ్ళీ నీటికి సపరేట్గా ఇప్పుడు ఒక్కసారి మెన్షన్ చేసుకుందాం. మరి ఐపీకి కూడా ఒకసారి చూద్దాం, యాన్యువల్ ఎగ్జామ్స్కి కూడా. మరి ఈ ప్రోడక్ట్స్ అన్నీ ఎలా గుర్తుపెట్టుకోవాలి అంటే చిన్న హింట్ ఉంది. ఈ హింట్ కూడా మీరు జాగ్రత్తగా అంటే హెల్ప్ అవుతుంది. ఓకే, రైట్. వాట్ ఈస్ దట్? ఇక్కడ ఒక్కసారి చూస్తే రైట్, ఫస్ట్ వన్. ఓకే, జస్ట్ ఫర్ హింట్, then remember this all the products in Krebs cycle. Chili chicken. What is that? Chili chicken is over. Kalyan send some fried mutton and onions. Okay, this is small hint for remember this total products in the Krebs cycle. Okay, right. Here in chili, the C stand for citric acid. Okay, in chicken, C stand for citric acid. Okay, is I stand for, that is isocitric acid. Okay, in over, O stand for, that is oxaloacetic acid. Okay, in Kalyan, K stand for ketoglutaric acid. Right. In send, S stand for succinyl coenzyme A. Okay, in some, S stand for succinic acid. Next, in fried, F stand for fumaric acid. Okay, in mutton, M stand for malic acid. In onions, O stand for oxaloacetic acid. Okay, right. So this small hint get help for the total number of, okay, just remember that product. Okay, right. Next, here neat. Okay, totally In mitochondrial matrix, okay, one turn of Kelvin cycle, okay, the one turn of Kelvin cycle, Kelvin, not Kelvin, Krebs cycle, okay, the one turn of Krebs cycle, then liberation, the 12 moles of ATP. It is very, very important for objective, okay, right. The one turn of Krebs cycle, okay, one turn of Krebs cycle, one turn of Krebs cycle, then synthesize 12 ATP, 12 ATP, okay now, right. Then the two turn of Kelvin cycle, the two, Kelvin, Krebs, the two turn of Krebs cycle, then produce 24 ATPs, 24 ATPs, okay. Then one turn of Krebs cycle, one turn of Krebs cycle, then release CO2 number, release CO2 number, okay na, release CO2 number, that is 2 CO2, okay. The two turns of Krebs cycle release CO2, 4 CO2, right. Then the total number of ATP produced in the Krebs cycle, that is 24, okay, right. In this Krebs cycle, which are in participate, okay, which are participate molecules and which are released, okay. The acetyl coenzyme A, how many acetyl coenzyme A are required here? The 2 moles of acetyl coenzyme A required here, okay. In 2 acetyl coenzyme A, out and release, okay. Release ఒక్కసారి చూడండి, ఇక్కడ 2 acetyl coenzyme యూజ్ అయినప్పుడు ఎన్ని coenzyme Aలు బయటికి వెళ్ళాయండి? 2 coenzyme A, okay. The 2 moles of coenzyme A is out, the 2 moles of coenzyme A is in, okay, right. Next, OAE, how many, okay, the Krebs cycle is begins with 2 moles of acetyl coenzyme A is condensed with the 2 moles of oxaloacetic acid, okay, right. 2 moles of oxaloacetic acid, right. Next, the producer number of oxaloacetic acid are 2 number, right. Next, how many number of ATP is produced in the Krebs cycle? Okay, that is 2 number of ATP is produced in the Krebs cycle. How many required of ADP in Krebs cycle? That is 2 number of ADP required in Krebs cycle. Okay. Next, the total number of NAD plus required in, how many number of NAD plus required in Krebs cycle? Okay, that is 6 NAD plus is required. Here this is 2 NAD plus, then 2 NAD plus, okay, 4, then 2 NAD plus, total 6 moles of NAD plus is required in the Krebs cycle. Then the total number of NADH2 produced in Krebs cycle, right, same case, that is 2 NADH2, right, 2 NADH2, 4, then 2 NADH2, 6 NADH2 is produced in the Krebs cycle, right. How many number of FAD plus is required in the Krebs cycle, right, that is 7th step, okay, 8th step, that is 2 moles of FAD plus is required in the Krebs cycle, right. How many number of FADH2 produced in Krebs cycle? That is 2 moles of FADH2 produced in the Krebs cycle, right. How many moles of water is required in the Krebs cycle? Okay, the total number of water molecules in Krebs cycle 8 moles of water is required. Okay, then the 10 moles of water is liberated, release, eliminated. Okay, next the total number of oxygen in the Krebs cycle, that is 4 number. Then total number of CO2 in the Krebs cycle, that is 2 moles of CO2 and 2 moles of CO2, that is 4 moles of CO2 is released in the Krebs cycle. Okay, it is very important for both objective and in annual exam also. So please subscribe my channel, okay, with the bell icon. Okay, thank you students. Have a nice day.